All right, guys, thank you so much for coming. So today we are on our second lecture. We're going to talk about basic chemistry. We're pretty much going to talk about how the elements react in the body and the elements, compounds and molecules in the body. And then we'll talk about the four organic compounds in the body, all right, which include proteins, carbohydrates, lipids, and nucleic acids. The nucleic acids are important because they hold the genetic, information which is important for reproduction and continuity of the, of the organisms. All right, and then we'll pretty much talk about how reactions in this body are regulated by the enzymes, which fall under the protein group. All right, and so that's it. So I'm going to assume a lot of the basic chemistry knowledge, such as you all know what matter is, right? What is matter? Is it too late in the afternoon? Anything that takes up space and has a mass. All right. So what is mass then? The amount of matter, yeah, in a substance. All right. And then weight. What would be weight? What's that?
Gravitational force.
Upon that mass. There's the pull of gravity on that mass. All right. And we know what molecules are, right? As opposed to compounds. What's a molecule? Any two atoms together, right? They could be the same form, like a hydrogen ion. Right? Hydrogen and oxygen, O2H2 molecules. But compound is when you have two different elements in the chemically combined. Okay, guys? So I'll talk about the properties of the ionic compounds and some of the bonding, the chemical bonding that occurs. So each element, of course, has got unique characteristics, all right, physical, that actually make it a unique substance, right? And then there's two types of these characteristics. The physical characteristics is what your eye can detect, what you can see. So that means the physical characteristics that you can see. What part of the study is it? Is it anatomy or physiology part? The physical properties. What's that? Physical properties. What you can see with your eyes is the anatomy part. You got fooled by the physics. Okay, so that's the anatomy, right? What you can see, what you can feel, and the characteristics is anatomy of the this is the anatomy of part of the study. And then this one, chemical properties, talking about how these elements react together, right, and interact with another. So what does that mean? That's the physiology part. So today we'll talk a lot about the physiology part in terms of the organic compounds and their functions in the body and what they do. All right? Did you get one? Oh yeah. I thought I counted you. Oh, you walked in just after I had given these guys. Oh, I'm sorry. There you go. So, anatomy and physiology. So we'll talk a lot about the physiology part of things today. All right? In this lecture, at least. For the elements. So the human body is made up of all these elements, right? Including carbon, hydrogen, oxygen, nitrogen, all right? And then some elements that occur at about 4% of the body, and then trace elements that occur in minute quantities, less than 1%. Okay? So in the four organic compounds that we're going to talk about today, the bulk of their makeup is these ions, the ones that make up 96% of the body. All right? Carbon, oxygen, hydrogen, and nitrogen. All right? And then, of course, these elements are the ones that required in less amounts, but 4% of the body is made up of these. All right? Phosphorus, which of those organic compounds contains phosphorus? The lipids, some of them have, Yes, yes, phospholipids, yes, lipids, they contain that, all right? And also genetic material, nucleic acids, remember, the phosphate backbone. Okay, so that's good. What about sulfur? Which of those four contains sulfur? Which of those four organic compounds contain sulfur? Proteins contain sulfur. Okay, some amino acids like cysteine and so forth, and magnesium, and calcium, and sodium, and potassium are important for electrical conductivity in the neurons, right? We'll do action potentials later on, and chlorine is also found in blood. Iron is, of course, for hemoglobin, right? And then trace elements are the ones that are needed in very minute quantities, and they're usually part of enzymes. They kind of catalyze or affect the rate of the enzyme reactions, all right? And things like selenium, all right? Manganese, copper, all right? And zinc. All those elements are trace elements, and they're usually part of enzymes, all right? So the bulk of the body though is up here. And we'll talk a lot about those. In fact, when you start talking about those compounds, those organic compounds, we'll be using them up and then adding on those minerals that are okay with you. Alright, so chemical bonding. The first type of bonding we're going to talk about is ionic bonding. Alright? And ionic bonding occurs when atoms either lose or gain an electron, or two, or even three sometimes. Alright? So when an atom loses an electron, what kind of compound is it? Lose an electron, what do you get? What do you call it? This is one of the things we have problems with in the LS modules. When you lose an electron, it's a cation, yes, because then it's positively charged. Because an electron is negatively charged, so it's a cation. And then, of course, gain electron is going to be negatively charged. It's called anion. Okay? So anions, of course, are attracted to cations and form ionic bonds. All right? And these are arranged in a specific lattice, like sodium chloride, for instance. All right? Chloride is the anion. The sodium ion is the positively charged cation. Right? It forms well-arranged molecules, right, in a lattice. And then the other bonding is covalent, right? But what happens in covalent bonding? What is a covalent bond? Yes, the atoms are sharing electrons. Okay, and there's two types of covalent bonding, right? I'm talking in terms of how the electrons are shared. Well, that's true, yeah. But I'm talking in terms of that. But that's true too. The single and double means single, one pair is shared, double, two pairs are shared. But what I was referring to is the polarity, because sometimes those electrons are not equally shared, and then the compound is polar. All right. If they are equally shared, then it's a non-polar compound, right? The reason I was talking about that is because of this. That's how you get hydrogen bonding in polar molecules, like water, right? This hydrogen bonding occurs when hydrogen is bonded to oxygen, or sometimes it's nitrogen. And water is a big example of the hydrogen bonding. So what happens is that oxygen needs 2 valence electrons because it's got 6 in its outside shell, need 8, right? So it's got a greater affinity for electrons. So when hydrogen is involved, what happens is that the electrons tend to be pulled towards the oxygen, right? So that this gives the oxygen a slightly positive and slightly negative charge. And then, because in fact, hydrogen is only a proton, no neutrons, with an electron around it. So, that's why when hydrogen loses, an hydrogen atom loses an electron, it's called a proton. Okay? Because that's all there is. So the one electron that's here is more attracted to the oxygen here, more attracted to the oxygen. And then, so therefore, you have on the hydrogen part of the water molecule a slightly positive charge. So the attraction between the slightly positive on the hydrogen and the slightly negative on the oxygen, that's what creates the hydrogen bond. Alright? So I want you to be able to explain this hydrogen bond. Like in an exam, I'll ask you about the hydrogen bond. So that's how we explain it. And it only occurs when hydrogen is bonded to either nitrogen or oxygen. Okay? Alright, so now we're talking about the kind of reactions that, you can either have a synthesis reaction or a decomposition reaction. Right? So a synthesis reaction, what happens in a synthesis reaction? You have smaller compounds or smaller elements or smaller reactants that are joined to form a much more complex and larger product. You always have, whenever you have a chemical reaction, you have reactants and product. Right? And then the one that you can reverse from product to reactants is a reversible reaction. Right? So, for instance, let's say you're joining two amino acids. You can form a dipeptide and you lose water. So it's a dehydration reaction. Synthesis reactions are dehydration reactions, and decomposition reactions are hydrolysis reactions. You break down by adding water, and you create by releasing water. So that's what it is, right? Because you lose the OH on one and the H on the other, forms H2O and lose water. So a synthesis reaction is also a dehydration reaction, all right? Because you lose water, all right? And then now we decompose, we're going the other way. It's a hydrolysis. Hydro is the water, lysis is splitting. So it's like you're adding water to split. That's what it is. Hydro is the water part, and this is to split. This is the H2O. So break down hydrolysis. You add water to it, like a disaccharide, for instance, and then you form the two glucose units. In fact, one of the problems you had with the LS module was the disaccharides as well, in terms of which identification of disaccharides. So this would have been which disaccharide when you have two glucose molecules. I know it's not a very common one. Huh? Somebody said it. Starts with an M. Yeah, maltose. Yeah. Or did somebody say it, or I heard it? Wishful hearing, wishful hearing. But yeah, that's that. And so that's the disaccharide here. And when you have fructose and glucose, then that's sucrose. Yes, fructose and glucose is sucrose. That's where you find in the fruits. And then galactose and lactose would be lactose. Right? Lactose in milk is made of galactose and glucose. So galactose and glucose. Galactose. Okay, very good. Moving on. So now we're going to talk about factors that actually influence rates of chemical reactions, and we're also going to talk about why that is. All right. Okay, so concentration affects the rate of a chemical reaction. The higher the concentration, the faster the reaction, right? Why is that? Why is it when you have a highly concentrated reactants, the reaction goes faster? Yeah, you're on the right track. What needs to happen for things to react? What happens? What really needs to happen when things are to react? If you want to make love to somebody, you have to be with them, right? You have to come together. You know, same thing. If the molecules are going to react, they have to bump into each other. They have to collide with each other. So if there's more of those reactants, there's a greater chance of them bumping into each other, right? Make sense? So when you have higher concentration, then the reaction goes faster because the likelihood of them bumping into each other is much greater. All right? So in the exam, I'll ask you about telling me the factors that affect the rate of chemical reactions, and I'll ask you to give me the or to explain it. Didn't get too far, right? Okay, so temperature also affects rate of chemical reactions. The higher the temperature, the higher the rate of reaction. Why is that? The molecules are moving faster, and the chances of colliding is much greater. That's why we have speed limits, right? It's now 70 on the intercept, on the highway. You know, if it's higher, the likelihood of guys bumping each other becomes even greater because the reaction time is even shorter because you react and you're ready to go. So that's why we have speed limits. Because the faster we are going, the chances of actually us bumping into each other are much greater. Just the same with the molecules. The faster the molecules are moving in that reaction mix, the more likely they are to bump into each other and react. So temperature means they are moving faster, and here it's just the likelihood, the probability of them bumping into each other is much greater. And here they're moving faster, so they get to each other quickly. Right? And a catalyst. A catalyst would fall under which one of the organic compounds. Catalysts are what? Proteins, yeah. All the catalysts, all the enzymes are proteins. And the important things about proteins is what? I mean, in terms of enzymes, the enzymatic action, is that it must itself not be depleted or not be changed, okay, for it to be a successful enzyme to catalyze a reaction. Why is it necessary? Why is it important anyway? That the enzyme itself is not changed and the enzyme itself is not depleted for it to be a good catalyst. Okay, what can happen if the enzyme starts to change? If the reaction changes the enzyme, then in no time you will, let's say the reaction denatured the enzyme. Why is that not good? Because then it won't be able to do what? Yeah, it will be slow, one, because then, because remember enzymes are specific for one. They only have this lock and key enzyme, so, if you change the shape of the enzyme, it will no longer be the catalyst for that, right? So it shouldn't be changed, and if it's depleted, then in no time you have no enzyme to do the reaction. Then it will only work for a little bit, and then after that it quits. There's no more enzyme around. So it must not be depleted, and it must not be changed. So it's very important for a catalyst, without itself being chemically changed. So enzymes are biological catalysts that are proteins. Okay? So that's the concept when you are explaining the reason or the way catalyst affects the rate of reactions. You have to talk about the effect that they must not be changed, they must not be depleted. Because if they're depleted, then soon enough there'll be no enzyme to do the reaction. Okay, so particle size. The finer the particle size, the smaller the particle size, the faster the rate of reaction. Why is that? Okay, again, it's got to do with the contact. A small particle is greater. I mean, the surface area to volume is it's almost one. You know what I'm saying? But a bigger particle, the volume is much greater to the surface area. So only a small part of a bigger molecule is exposed to the actual reaction. It's like when you have a small ice block like this and a much bigger ice block. if you pour the boiling water on top of the big ice block, it will take a long time for it to get to the inner ice. So the reaction is going to be slow in terms of melting the ice. But if it's fine, it's small, when you pour the water on top of it, you almost cover the volume as well. So it probably just melts in one time. So that's how small particle size affects reaction rates of reaction. The finer the particle, the smaller it is, the faster the reaction, because the area that is exposed, you know, in relation to the volume is a lot greater than a bigger particle. And also the chances of them colliding is much greater too. And they move faster in solution too. The bigger it is, the slower and kind of clumsy it's going to be to move in the solution too. But for particle size, the unique thing about particle size is the area that is exposed on that particle. It's much bigger for a smaller particle in relation to the volume than it is for a bigger particle. Okay, so use all those reasons when you are explaining the effect of these factors on rates of reactions. Because I will ask you not only to tell me the factors, but to explain them. Okay? All right, so we start off with organic compounds. All right, so you know, these are unique to living systems, and there's 4 of them, like we said: carbohydrates, we get lipids, and they all have different functions. At the end, I'll give you a table just summarizing all the functions of these organic compounds, right? And then, of course, proteins and lipids and genetic material. And you will see as we move from carbohydrates to lipids, we start adding those elements. We start off at the top, going down, and adding all those other elements in these compounds. And the nucleic acids have most of them, most of the elements that we talked about. All 4 up there, and then also phosphorus and nitrogen as well. Okay, so we start with carbohydrates. Okay, so first of all, let's talk about the word carbohydrate. What does it mean? Okay, we get carbo. What would that stand for? Based on the elements we talked about. Carbon. Okay, so this is carbon. So hydrate, what does that stand for? What do you think of when I say hydration? What? Okay, so carbohydrates are carbon with your water molecule in terms of the ratio. So the ratio we said is 1 to 2 to 1. Carbon, hydrates, H2O. So that's why we call them carbohydrates. All right. All right. So now, and then of course, I think you also had a few problems with, you know, the polymers that arise from adding more units of these glucose units. So the major function is to supply cellular food. All right. In terms of which carbohydrates do we use for cellular food? The most common? Yeah, glucose. And in fact, the brain will only use glucose. It will not use anything else. That's why people who are diabetic, when they cannot, control their glucose properly, sometimes they go into a coma. Because the brain will only use glucose. It doesn't use anything else. Other parts of the body, other cells can utilize other carbohydrates and so forth, but the brain only prefers glucose. All right? So, we get the monosaccharides, glucose and fructose, right? Which we said make up which of these disaccharides is made up of glucose and fructose? Sucrose, right? And then of course lactose, we said is made up of which and which? Yes, galactose, and in the morning we call it G plus G. The other one is galactose, GL. So glucose plus galactose is lactose, and then of course 2 glucose molecules equals maltose, right? So polysaccharides would be many glucose units, and that's starch and glycogen. And glycogen is stored, 1, in muscles. Where else is glucose stored? In the liver, right? Why do the muscles store glycogen? Yeah, for energy. They use it for energy. Especially the white muscle fibers, they break down glucose with no use of oxygen, and you get especially athletes that sprinters, they have more white fibers, and more white fibers are much bigger in terms of diameter. Why? Because of storing glycogen. All right. The red muscle fibers, because they use oxygen for the energy, which is oxidative phosphorylation, they are thinner in in diameter. So white muscle fibers are thicker because of the storage of glycogen. Right? Right. And another thing I wanted to mention about carbohydrates, they not only the cellular energy, they also form they are on the surface of cell membranes attached to the proteins, and they form glycoproteins, which are very useful for cell recognition. And they are also called cellular adhesion molecules. Cellular adhesion molecules, CAMs. Cellular adhesion molecules. Okay, so for instance, when a white blood cell is going to the site of injury, it has to stop there, and then it has to be anchored there. So there are those molecules that are on the surface, which include a carbohydrate, that will anchor that white blood cell in place because the blood is moving, right? So it will anchor it there, and the integrants will kind of sort of lead it to the site of injury. Okay, so carbohydrates take also part in cell-to-cell recognition, and it affects the immune system a lot. Oh, what did we talk about this? Yeah, this is our little sucrose right there, glucose and fructose. And of course, what kind of reaction is this? It's a synthesis reaction, so it's a dehydration reaction because we lose water, right? So that's a synthesis reaction. Okay, very good. Okay. Now we go to lipids, right? And lipids are non-polar, so they don't dissolve. They don't like water. They're hydrophobic. They dissolve in things like non-polar solvents like acetone and alcohol, right? One of the questions that came up in the module was talking about fatty acids, and it was asking me to describe a fatty acid, pretty much it said what is a long chain of carbon with carboxyl end, and carboxyl end is simply COOH. So this is the carboxyl end of it, and so a fatty acid would have all that, and this is a so fatty acids. It's called an acid wine. Why is it called an acid? Because it can give off a what? What's an acid? An acid can give off a hydrogen ion, right? It can give off a proton if you want. So this can give off this hydrogen ion, all right, and you're left with COO minus, like this. See what I'm saying? So that's why they're called fatty acids. And they've got this long chain of carbons. And one of the other questions that came in the module was like, what are the two differences? What are the two factors that show differences between fatty acids? Right. One of them is this, which is the length of the chain. Will show the difference between some fatty acids are longer, some are shorter. Okay. The other one is, does anybody know how the differences between the fatty acids? I said one is the length of the chain. The other one is the kind of bonds that occur in the chain. What are some of the bonds called? Some are double, yes, and so it's an unsaturated fat. And the other one is a single bond, which is saturated fat. So that's the differences between fatty acids. That I think people have trouble with that in the module, and I will cover it again towards the end when we finish off talking about this. So we have C, H, and O in the fats, but not too much O. And not as much O as we have in carbohydrates. All right. Okay. So what are the examples? We got the fats or triglycerides. Usually they store energy. They store twice as much energy per unit weight than carbohydrates. That's why if you take a lot of carbohydrates, they will be quickly converted to fats. All right. So what is a triglyceride? It's 3 fatty acids on a glycerol backbone. Glycerol has got 3 carbons on it, and then you get H's on it. All right. So it will have fatty acid number 1. All right. And then fatty acid number 2. Oh, I love the color somewhere. Fatty acid number 3. And they could be same or different lengths. Okay, which is how the differences come about between fatty acids. So it's called a triglyceride because of the glycerol backbone. Right? Phospholipids are modified triglycerides because they have a glycerol with 2 fatty acid chains on it and phosphorus. So they're called phospholipids. And where do we find phospholipids? Structural components of the plasma membrane, right? Okay, so we got that fats, phospholipids, and then eicosanoids are like 20 carbon, molecules that affect physiological functions. They are hormone-like substances, like prostaglandins, okay, that affect smooth muscle, okay, because it contracts. In leukotrienes, which are very active in the immune system, they activate immune cells, right? So those are 20 carbon compounds. And then steroids. Steroids are 4 ring structures based on cholesterol, all right? So they got a ring like this, okay? Okay, and then this one is like a 4. The differences come about the groups that are attached here, 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 and there. But that's the backbone of it is cholesterol. And what are some of the important steroids? Steroids. What are some good examples of steroids? Okay? Yeah, cholesterol is the mother of all those steroids, and they everything is built up. But yes, for men, and estrogen for women, and at the end also. So for reproductive function, they control testosterone for men. They all come from cholesterol. Okay, and there are very minor differences between cholesterol. Between estrogen and testosterone. Okay. All right. So we start with fats. All right. Fats are also found in subcutaneous tissue. As subcutaneous tissue, which is under your skin. What's the function of subcutaneous fat? What is it for? Insulation, so that you can retain what? Heat. Yeah. And also for cushioning too. Like behind your eyeballs, to cushion it against the sockets of your eye, because the skeleton is hard bone. So cushions your eye. And what else where else do you find fats? Around what? Organs, yes. Very good. Organs, you find it around, no matter how thin you are, how lean you are, you have fat around your heart, protecting it, fat around your kidneys. Okay? So protection, insulation, and of course, energy storage. So that's the fats. And then phospholipids, of course, we talked about it, 2 fatty acid chains, plus that phosphate head, which makes it good for the plasma membranes. Right? And then, of course, steroids, which are based on cholesterol. And all these fat-soluble vitamins A, D, E, and K, all those. And hormones, like testosterone, we talked about estrogen, progesterone, all those are based on cholesterol. So the mother compound is cholesterol. All right? And then there are also some adrenal cortex hormones that come from so where are these made? Adrenal cortical hormones. Adrenal gland on top of the kidneys, yes. All right. So, and one of the biggest ones that comes from here, of the steroids that come from there, is called cortisol. Cortisol mobilizes glucose metabolism and all that. right? So cortisol is also a stress hormone. You know, when you're stressed, you release a whole bunch of cortisol, and then what happens if you're continuously stressed and you're releasing cortisol, you start to get fat, because the function of this is to mobilize a bunch of glucose. See what I'm saying? So that's why sometimes if you're stressed, you gain weight. Have you seen that? Because of the cortisol. If you're releasing too much cortisol, and for men usually they get fat around the bed. No, not six pack anymore. So if you don't want to be, if you want a six pack, don't be stressed. Actually, the thing about stress is that you can go to the gym and you can relieve stress that way too, when you're working out. So it's actually good, and it works your heart as well, cardiovascular fitness and so on. Right? So eicosanoids are those 20 carbon compounds I was talking about. Prostaglandins are hormone-like substances, but they're not quite like hormones. So they regulate all the metabolic activity in the body. Glucotrients, I was talking about this, important in activation of immune cells, and we'll do this in AMP2 when we talk about immunity. And then thromboxans. Anybody know what thromboxans are involved in? Does anybody know? Thromboxans. These are involved in clotting. Thromboxans are involved in clotting. All right. And then of course fat soluble vitamins A, D, E, and K. Fat soluble. All these are lipids. All right. And lipoproteins are used to transport fats in blood. So one of the good lipoproteins is what? High density lipoproteins, right? And why are these better than low density lipoproteins? It's to do with the protein, right? High density lipoproteins have more protein in them, all right? So that means they are more what? Soluble or insoluble? They are more soluble. The low density lipoproteins tend to cause cardiovascular problems because they are not as soluble, so they tend to settle in blood vessels and clog them up. Okay, so high density lipoproteins. So when you go to the doctor and they do a complete blood count, and they give you a CBC, complete blood count, and they'll do the lipids, they'll do HDL and LDL. If your cholesterol is low, but your LDL is very high and your HDL is low, it's still no good because of that reason, because the LDL can cause problems with that. All right? Okay, so triglycerides, the fats, we talked about it, glycerol backbone in pink, and of course this is a synthesis reaction, dehydration. Okay, use 3 water molecules because you get 3 fatty acid chains on there. They could be different in length. They just show they're equal. It's a diagram, and then just for diagram purposes. Is my thing working? I hope it's recording. Anyway, let me know if it's recording. It's got good sound on it. When you listen. Composed of 3 fatty acids, triglycerides, right? And then of course we'll finish off talking with, about the unsaturation, which is the second way fats can be different, right? This was in your module. So palmitic acid, which is a saturated fat, you find it in beef, pork, olive oil. Okay, and then some of them are monounsaturated fats like peanut oils and so forth. But the best kind, of course, is the polyunsaturated, which comes from fish oils, sunflower oil. Okay, linoleic acid is an example. You see, it's got multiple unsaturated points, and these tend to also clog your blood vessels. So that's why I say yes, beef and things like that, which is hard to do. But these are the best. So fish oils and sunflower oils, corn oil as well, very high in polyunsaturated fats, which are better for your diet than the others. Right? Okay, so now we go to phospholipids, which are parts of the plasma membrane. So this is the hydrophilic part, which is the water-loving part. Okay, and this is the hydrophobic non-polar chain. right? It's just carbon-carbon bonding, right? So on a plasma membrane, you've got this arrangement here. So what's important about that arrangement? Why is it that the heads are on the outside and the tails are on the inside? It's based on the properties of the head and properties of the tail, right? Yeah, and why would you want to expose the hydrophilic end instead of exposing the hydrophobic end? That would be better the other way. Where are these cells? What kind of environment are they? It's in an aqueous environment. Extracellular fluid is an aqueous environment. Cytosol is water. It's in the intracellular on the inside. So that's why this will be the inside of the cell also exposed to water. The cytosol is made of water. Outside of the extracellular part, it's extracellular fluid. It's all aqueous environment. So that's why you expose the heads on both sides. That's the reason why. All right. So that's the plasma membrane right there. Like modified triglycerides, because it's really got that glycerol backbone, but instead of having the 3 fatty acids, it will have the 2 fatty acids and the phosphorus added on it. So it's a phospholipid, right? And I think this was also in your module when it was asking about phospholipids, and it was saying, okay, you have a modified triglyceride with 2 carbon chains. What do you have? Most of you kind of got the phospholipid part wrong. Okay? But that's good, because if you keep doing it, you end up getting it right and you get your 5 points. So that's the good thing. That's why I like it. That module. And then the 20 carbon eicosanoids, also in the membrane, they also act as regulators of metabolic reactions, right? And then the steroids finally will also reproductive physiology also, and even other parts of the physiology like glucose metabolism and so forth, right? So that's the steroids. Okay, there you go, including cortisol and all those other things. So we move on to the proteins. These are macromolecules, which means they're very big molecules. And another thing that we kind of had problems with was grouping the proteins in terms of the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. Okay, so when you have the primary structure is really just the sequence of the amino acids, right? And then the secondary structure just includes simple folding, where when you have the hydrogen bonding, the hydrogen bond attraction. Okay, so you have a simple folding, and then tertiary structure, which is the third level, you have more complex folding, like secondary folding, and you are starting to involve the R groups of the proteins, which we'll talk about in a bit. Because the R groups on a protein will convey the attraction of the molecules, or the attraction of the molecules that are sticking out of the protein chain. And I'll show you that in a little bit. All right? And then the quaternary structure, the final structure, is when you have several folded proteins acting together. Like if they say there's 3 proteins acting together, then and folded proteins interacting. That's the quaternary structure, the 4th level of interaction. Right? So again, like we said, we've got CHO that we've been talking about. We've got N. Now we've got sulfur. We're now beginning to go down on those elements that are found in lesser, quantities in the body. All right. So amino acids. There's 20 amino acids. It's pretty much based on the R group. The R group is different, but the backbone is the same. So you have the amine end, right? And then you have the carboxyl end, or H. Okay? And then you have the H. So what do you have in an amino acid? You've got the amine end, and then you've got the carboxyl end, and then you've got the hydrogen. the R group is here, right? Okay, so that's what makes them different. And the simplest one is when this R group is hydrogen. You form glycine. That's the simplest you can get, right? Sometimes it's CH3. This R group is CH3, and you've got alanine. Okay. So, and then there's pretty much 20 basic amino acids. All right. So the same thing like we did with the sugars. If you've got 2 of them, it's a dipeptide. If you've got with the peptide bond, again, it's also a dehydration reaction. You lose water and they join up. All right. So, and then tripeptide, you have 3 of them, and then polypeptide, you have as many as hundreds of them form a protein chain. All right. And so the tertiary structure is influenced by how these R groups attract each other. That's why the sequence of these amino acids in the protein chain is very important, because it will juxtapose certain R groups next to each other. Okay? So if that is wrong, that order of the R groups is wrong, because the order of the amino acids is wrong, then the protein is not going to fold properly. And if it doesn't fold properly, it's not going to have the proper function, especially if it's an enzyme. It's not going to fit whatever the product is. So it has to have these R groups arranged properly so that when the protein folds up on itself, it's going to fold properly. Because these are the R groups that attract each other. Okay? All right. And then, of course, you get hundreds of these amino acids. Okay, so these are the amino acids, the building blocks of proteins. So this is pretty much that backbone I drew on there. And the 3 things that are included in an amino acid. So we have the amine group, right, which is the NH2. That's the amine group. And then the carboxyl group, which is the COOH carboxyl group. And then, of course, the hydrogen atom, the one on the bottom here. Okay? And then a side chain designated by R, and it could be as complex as for the tyrosine, which is a benzene ring on it. All right? Okay? And some of them have sulfur, in them like cysteine, is an amino acid with sulfur. Methionine, another amino acid with sulfur in the R group. And this tyrosine is important in the thyroid, where it combines with iodine. All right? And this stuff affects the metabolic rate of patients in the body. Right? So tyrosine is the amino acid that makes tetraiodothyronine, triiodothyronine, which are these tyrosine molecules attached to 3 or 4 iodine molecules. This is the one that appears 4% in the middle of those elements we talked about at the beginning. All right? And we'll talk about the tyrosine later on when we talk about the endocrine system, which will cover in exam 2 material. All right? So proteins. So proteins, what are the functions of proteins? We've already kind of talked about one already, such as the enzyme, speeding up chemical reactions, catalyzing chemical reactions. We already talked about this regulation of metabolic reactions in terms of the enzymes. And they can also be structural, such as collagen. Collagen is a fibrous protein, right? The earlobe is made of elastin, which is kind of like a stretchy protein that stretches, all right? Responsible for muscle contraction. We'll do that in a little bit, in a few lectures ahead, like myosin and actin. These are fibrous proteins that slide up upon each other like this when the muscle is contracting. So proteins are also very important for muscle contraction. All right. And then, of course, fibrous proteins they have strand-like appearance, such as those muscles for contraction. They are long proteins, long chains that slide up on each other. And also collagen. Collagen strength also happens when these long fibers are arranged in a very well-orderly fashion. It gives it strength. Because if they're all pointing like in one direction, it's like when you take 3 pieces of rope and or whatever, or thread, and you double them up, and they're all in this oriented in the same direction, it's going to be much stronger. You can prove it yourself by taking, sewing thing, the thread you use for sewing, and you take one and you try to pull it apart by itself. Then if you double it up and you try to pull it apart, you probably won't be able to break it. That's the same concept with the fibrous proteins in collagen. They are so arranged nicely in the same direction. That gives it that strength. Okay? And then globular proteins tend to be enzymes almost all the time. Why? Those factors we talked about at the beginning that affect the rate of reaction. We talked about particle size. We talked about moving speed. So if you have a very bulky enzyme, it's not going to be moving fast. It's not going to really affect the rate of reaction. If it's small and globular and it can move in, and then it will affect the rate of reaction. So all those reasons that we talked about at the beginning that affect rates of chemical reactions will be the reasons why you have globular proteins are more suitable as enzymes than maybe any other fibrous proteins, for instance, which are more bulky. And with the primary thing is for them to have strength. But here they are globular. Like antibodies, what's the other name for antibodies? Immunoglobulins, because they are globular proteins. They have a specific shape though, which is like a wine shape, but they are globular proteins all in all. Hormones are also globular because they affect rates of reactions, and of course enzymes. They are globular proteins. Alright? And then denaturation. When you denature, what are you doing to the protein? You are changing the structure of it. That's what you're doing. What you're doing is, remember those stages of arrangement. We talked about primary all the way to quaternary. When you denature the protein, you're pretty much breaking those attractions, like especially the hydrogen attractions. You do it in the morning when you make breakfast. If you eat the eggs, what happens when you fry an egg? It first off starts as a colorless fluid, right? A transparent fluid. Then after you fry an egg, what happens? It's white because it's denatured. Okay. And somebody in the morning was asking, okay, if you denature it, do you reduce the amount of amino acids? No, because you can't destroy or create better. The only thing you do is change the structure, but you still have the amino acids there. They're just not folded up the way they should be. Okay. In fact, it's better for you because it's like partially breaking it for you. When you digest it, it's better. That's why even meat, when we eat meat, we cook it first. If you eat raw meat, you might have problems digesting it. You see what I'm saying? Carnivores have a different digestive system that's adapted for digesting raw meat, like the lion and so forth, right? You can eat sushi and stuff like that, but fish, but we're not adapted to digest protein when it's not denatured first. Gotta denature it, and then you disrupt those hydrogen bonds and all those. That gives it shape, and it's non-reversible. It becomes non-functional after that. Once you change the structure of it, then it's not going to function the way it should. Just like when you have a mutation and you have the wrong sequence of amino acids in a protein chain, it's going to fold up differently. And when that happens, that protein is not functional. It's not functional. Okay, it's not going to work. So sometimes when you have a problem with your genetic material and it's not producing the right proteins for whatever part of it is, they will try to insert where they have genetic therapy, where gene therapy or something, they will try to insert the correct gene into your cell so that it can be expressed. Okay? They could use a virus to infect the cell with the gene. Okay, because viruses, they cannot multiply by themselves, so they need another cell to do it. So the way they do it is they insert their DNA, or RNA, if that's what it is, into the DNA, and then the cell will start making the DNA. So what scientists do is they take that gene that's problematic, that's been mutated, and insert it into the virus so that the virus infects those cells, and then they will start to express it. Kind of clever way of, it's very interesting actually. Anyway, so denaturation is no longer active, and it's not functional. All right, so characteristics of enzymes are kind of related to those qualities we talked about. These are functional characteristics because they have a function in nature. These characteristics speed up chemical reactions. That's what they do by lowering activation energy. I think your module asked you about activation energy too. The amount of energy you need to start a reaction is the activation energy. I think it was in the module too. And you kind of have some trouble with that. So the activation energy is the energy that you need to start off a reaction. So enzymes, what do? They lower that energy so that you don't need a lot of energy just to start it. So the reaction goes faster by lowering activation energy. Most globular, of course, most are globular proteins that we talked about. Again, because of those qualities, factors that affect rates of chemical reactions, and they act as biological catalysts. Okay, and they're of course chemically specific. That's why the structure of it and the way it's folded up is very important. Because if it's folded wrong, then it's not going to attach to the correct substrate for one. Or the correct substrate will not fit in the active zone of that protein. So they have to be specific. Frequently named after usually the substrate. Like lactase will break down lactose into what and what? Lactase breaks down lactose into what and what? What's that? Maltose is another disaccharide, right? Yeah, galactose and glucose. Galactose and glucose. That's what your module does, right? That's the LearnSmart module will ask you the same question in a different way to see if you get it. So that's what I was doing, to see if you got that. Remember, you had problems with the disaccharides in your module. So I was now going the other way. If you break down lactose, you get galactose and glucose. So it will be lactase. Lact standing for lactose, which is the substrate that you're breaking down, and then ASE. That's the terminology. Acetylcholinesterase, same thing. It will break down acetylcholine, right? Usually the neuromuscular junction, because acetylcholine is a neurotransmitter, right? Okay, so chemical events in the body are regulated by these, enzymes, and the way you affect the rate of reaction is by affecting the concentration of the enzyme, right? The more enzyme you have, the faster the reaction. The less enzyme you have, the slower the reaction. So the body will regulate reactions in the body by regulating the amount of the enzyme, all right? Or the rate at which the enzyme is going to work. So these are the two ways by which you can control the enzymes, right? By concentration or by changing the activity of the enzyme. Okay? Sometimes enzymes have what is called the allosteric site, which is another part other than the active site that attaches to another molecule that will stop it. Usually it's the product of the reaction that will come back when you have enough of the product, will come and attach on the allosteric site on the enzyme to stop it, right? When you have enough product, right? So those are the two ways by which you can regulate enzymes, by affecting the concentration and the activity of the enzyme. So our final one is the nucleic acids, all right? And the nucleic acids are very important for what? For heredity, genetic material, right? So the DNA and RNA are made of nucleic acids. And it's composed again, you see we're adding all these compounds. MP now, with phosphorus. Alright? So you have three things, the nucleotides, that make up these nucleic acids. So what you have is a 5-carbon sugar called ribose. Alright? and also you have a phosphate, and then you also have a base. And a base could be a 2-ring or 1-ring, base. And we'll talk about that in a moment. All right. So you have those 3 things: the monosaccharide, which is in this case a ribose, 5-carbon sugar, 5-carbon ribose. It could be deoxyribose for the DNA and ribose for RNA. Okay, but it's got 5 carbons. You just remove an oxygen molecule from the ribose, and you have deoxyribose, which is used in DNA. Okay, so nucleotide is the basic unit, which is comprised of those 3 things. These are the bases I was talking about. Adenine and guanine have the 2 rings, like I drew up there, and usually called purines. They're not usually called, they're always called purines. Okay, and then the other ones are cytosine, thymine. These first 4 are in DNA, and we'll talk about that a little later. And then in RNA, instead of thymine, you have uracil. And all these 3 are 1 ring. Okay? And those ones with 1 ring, the bases are called 5-ring-mediates. Okay? So, these are the 5 nitrogen bases. And then, of course, DNA is deoxyribose for the sugar, and RNA is ribose. Ribonucleic acid, deoxyribonucleic acid for DNA. Alright? Okay, so DNA, we'll start with DNA and go to RNA. So I want you to also note the differences between RNA and DNA, right? So DNA is a double strand. A double strand where you have that phosphate backbone going down like this with the bases in the middle. Okay, so the bases will be the purines will always attach to a pyrimidine, so that the 2 bases and the 1 base will attach. Okay, if you have two bases, they're too long, it won't fit in the gap. If you have 1 base here, 1 base here, it won't fit again because they won't touch. So you always have, like, adenine always pairs with thymine, guanine with cytosine, or uracil in RNA. You see what I'm saying? Okay, so, and of course, DNA is the genetic material. It's simple nucleus. Alright? And it's locked up in the nucleus with a double envelope, and it's folded up, and it's an alpha helix. All the fact that it's a double-stranded had to do with one thing, is to protect it. Okay, RNA is single-stranded, and you can easily change the structure, but for DNA, because that's the blueprint of the body, so you don't want to have a mutation in there. And the DNA doesn't disappear, it's only strand, it's also attached to proteins, histones, and it's called chromatin, okay? Again, to protect it so that it doesn't get, mutated or anything. Okay, so it's double-stranded and it's for protection. Okay, so note when you're talking about RNA, the differences. And then, of course, it replicates itself when a cell divides, so that you have the exact same genetic material in the one cell and the daughter cell. Right? And it, of course, promotes genetic continuity, which is associated with which characteristic of life? Again, reproduction. You guys are fast. The other guys in the morning, maybe it was because they didn't have enough coffee. Yeah, provides instructions for protein synthesis. All right. So each cell is specialized to make certain proteins, or it will express certain proteins, and it could be a certain enzyme. All right, that has a specific function. So you have different cells in the different tissues that make up all these different proteins for the different reactions that occur in the body. Right? So each cell is specialized for making a specific protein. That means it has a specific function. Right? So again, like we said, adenine pairs with thymine, guanine pairs with cytosine. This is a purine, pyrimidine, purine, pyrimidine, right? 2 ring, 1 ring, 1 ring, 2 ring, right? Okay, so, and this is replaced by uracil. And the other difference, deoxyribose. Sometimes in the exam I'll say, okay, can you tell me the differences between DNA and RNA? Right? Then you start telling me about the double strand for DNA, you start telling me about deoxyribose DNA, you start telling me about thymine instead of uracil in DNA. Okay, 3 points. Alright, so ribonucleic acid is single-stranded, because it's also important that, because you can always make another copy of RNA, right? You can always make another copy of RNA, but the DNA has to be double-stranded so that it's well protected, so the bases are not exposed. It's composed of a monosaccharide ribose. Again, you see the differences with DNA, right? And then instead of thymine, in RNA, you have uracil. And I think this was one of the things you also had problems with in your module. I think when it was talking about DNA and RNA and nucleic acids, some of it. Okay? So those are the differences, not the differences. And of course, we get 3 types of RNA. Messenger RNA is one that does what? Transcription. It will take the code on the DNA and move it out of the nucleus. And the messenger RNA moves into the cytoplasm where it will attach onto a ribosome. And then transfer RNA, what does it do? It transfers amino acids. And it's got a 3 base codon on it, like ATT, for instance. Not the phone company. I didn't need timing. Okay. So and it will, each 3 base sequence will code for a particular amino acid. One of those 20 we talked about earlier on. Right? So each one will have maybe 3 or different kinds of codons. Right? And then each will have its own its own transfer RNA that will pick it up and pick the amino acid, and then it will drop it in the sequence along the messenger RNA that will match those 3 bases. And then of course ribosome RNA is found on the in the cytosol, and usually this ribosomal RNA makes proteins that the cell uses. The cell will use it, okay? And if the proteins are made in the endoplasmic reticulum, because the endoplasmic reticulum is a membrane-bound organelle, remember? So those proteins can be packaged and moved out of the cell. So usually the endoplasmic reticulum will make proteins that will be used by other cells. Okay. Whereas these ones are free-floating ribosomes, and it's right in there, the proteins is made and it's already the solution inside. So the cell will use it. Right? Alright, so there's that. And this one here just shows you the functions that we've been talking about throughout. Carbohydrates, of course, you've got them as the energy cell, and I was talking about cell adhesion and cell guidance, alright, on the outside of the cell, alright. Structure as well in the DNA and the RNA, ribose and deoxyribose. And then the lipids, they store energy well, right? And then the structure of the plasma membrane, phospholipids, right? Sometimes I may say, okay, what are the general functions of the lipids? What are the general functions of the proteins? And we talk about that. And all this we talked about, we didn't leave any of these out. This is just a nice table that summarizes all of them into one place, right? And then regulation, of course, those eicosanoids and so forth, the leukotrienes, the prostaglandins, all right? And also regulate tissue inflammation, right? Then insulation, of course, we talked about cutaneous fat under the skin, and then of course amino acids regulation, the enzymes, structure, we get collagen and all those fibrous proteins. Energy, actually you get as much energy as you do from carbohydrates from proteins. You know what I mean? Isn't that cool? So you can actually use proteins as carbohydrates. Usually if you want to lose a lot of fat, you do sometimes what they call the Atkins diet, where you eat a lot of protein. You won't run out of energy because proteins can be broken down to get as much energy as you would per unit weight as carbohydrates, which is good, right? It's just that carbohydrates are sweet. Most of the athletes, like right now, what are they called? LeBron and them, they are taking carbohydrates out of their diet and they've lost like weight. Have you seen LeBron lately? He's like a skeleton like this. You know how it is when these guys are tall and they get so skinny? He's like a skeleton. Yeah, he just cut the carbohydrates from his diet. All right, and then all that, and then muscle contraction and all these other things, and then of course nucleic acid is the genetic material. Okay, guys, thank you so much. We'll see you in a little bit. All right.